Okay, hi guys, this is chapter 20 and this is talking about biotechnology. And as you can see, there's so many things we need to talk about. But a lot of details are in the chapter, so go ahead and read the chapters. You need to read every single word. Even though if they don't understand, reread them, Google them, text friends, family member, or you can just go on Schoology and then send me a message and I can help you. So anyway, these are a few things that we need to know for this chapters. So let's get started. What you must know, I'm gonna pause a few seconds so you can, stop the video. So a lot of terminology is important, and we need to talk about some ideas that basically how can we use a lot of modern biological technology to help us to solve a lot of problems in our life. Okay, so genetic engineering, it's a process of manipulating genes and genomes. We got very good with this technique. And then in terms of biotechnology, it's a lot of different things that we can use to, for example, genetic engineering, and then how we can use the genetic information that follows under the biotechnology term. Furthermore, recombinant DNA. So what we can do is actually we can artificially make a new strain of DNA by combining different sources. So for example, we can insert human genes into E. coli, and then we can also clone genes. We can actually cut genes into small fragments and just copy that so many times so we can use it in the lab instead of, oh, we need more genes, let's cut a little bit of your skin cell off again. So some of the tools that we'll be using, these are really cool. Okay, restriction enzyme. Restriction enzymes are designed to cut a very specific site of a DNA. So you can actually design, I don't know, let's say there's a gene that has a length of, actually, let's use this sentence. Let's say the phrase tools of genetic engineering. Okay, I can design an enzyme that cuts at every single L between L and S. So right here, it would just cut, and then it would just be this side and that side. Or I can design a enzyme that will cut right after every single O. So it's cut right here, cut right here, cut right here, and that's it. Or I can design an enzyme to do a very specific job. So that's what restriction enzyme does. And furthermore, restriction enzyme cuts into small little fragment. But as I said, there's always a sticky end. Basically, that's an end of that only has one very special characteristic from this entire sentence. That's what sticky end is. Okay, but if we need to cut something, we also need to put something back. Well, DNA ligase is going to help us to do that. As you can see, it's a protein, and what it can do, it can glue everything back in a way. So we can even insert something. So for example, tools of, let's say if I want to insert a gene, well, a word right here, it says awesome genetic engineering. So I can use DNA ligase to kind of put it back, says tools of awesome genetic engineering, so on and so forth. And also we can also clone the DNA. So just like what we said, like it's very similar to what we talked about in chapter 19 when the viruses are kind of like cloning itself. So like that. So another thing that I want to show you is the picture of it. Instead of using just a sentence, I want to see like how it's done, well, in a picture form. So this is DNA. So we have the restriction enzyme here cutting right after G and then cutting everything in between it. So that little part will be what we call the sticky end. Just imagine that's kind of like the Velcro, the sticky end, the one that has claws, that one. Okay, and then now because we want to introduce something, so this is something new introduced in here, and then you can see it also have the opposite side of sticky end. One side has this sticky end, the other side has that sticky end, so they can fit perfectly. And once you fit perfectly, then DNA ligase will all go ahead and seal everything, glue everything back. That's how we do by doing the recombinant. Gene cloning. This is very similar to viruses. Instead, we are not really actually using a viruses at this moment. So basically what we can do is we can insert a gene into plasma. okay, right here. And then when you have a replication of that time, we have a recombinant of this genetic information in this bacterium cell. Okay, basically just insert something that's foreign into something that is, I guess, host. Okay, and the application of this is we can actually do a lot of things. We can actually create something, some special talent, if you will, to organism, or introduce that special talent to organism that didn't have that special talent before. So, for example, specific pest resistant gene for plants, special clog dissolving protein for hard treatment, and then different kind of treatment right here, as you can see right here. So, furthermore, by using genetic engineering, we also need to kind of figure out how to do all these kind of stuff. Well, some of the techniques are as follow. One of them is called PCR. Like I said, we can actually copy many, we can create many copies of DNA by using just one original one. Okay, so we don't actually using the cell to create more DNA. We could, but it would take a lot of more time and it would be more complex. It would be far more complex to do, and then you have to purify the DNA and so on and so forth. So this is a way to do it without using the cell, and you can make lots of copies. So furthermore, by making copies, we also need ability to analyze, well, how do we copy? If we copy it wrong, we need to know we did something wrong, and so on and so forth. So gel electron phoresis. This is something that you will use in college, and then it will be really cool when we have the lights shining on that gel. So I loved it personally when I was in college, but just random note. So what it does, it's actually use sample in the small little wells at one end of the gel, and you put a power source, usually like a meter kind of thing, that runs electricity through the gel. And what it does, it will attract different molecules and then align it according to its size. Okay, so we're basically pulling this information closer to one side versus the other side. Okay, so whatever it's shorter material, let's imagine it will have a shorter segment of the DNA, it's going to be on the positive side, and then longer segment of the DNA is going to be on the more negative side of this power source. So this is gel electrophoresis. Phoresis. I cannot say it right. I'm sorry. So it looks like that. Really cool. And if you do it right, you get to do a lot of these. So another thing is, well, we also have the technology to do cloning organism. So what we can do is we take, for example, in this one, we take an egg from this thing, and then we basically suck it out one of its nucleus. So it still have the cell, it just doesn't have a nucleus. And then now we fuse this nucleus into this the cell. Okay, so what it does, it allows the cell to do exactly what the nucleus is supposed to do, but in different type of animal. For example, we can introduce human genetic information into, in this case, sheep, a sheep cell. So this sheep cell will actually produce human useful protein, but in a sheep body. So furthermore, let's look at it. So like I said, nucleus from a mammal cell, so it has go through everything, and then it put it into a mummy, and then now you can have a birth of a baby lamb that would be identical with its memory cell donor. So whoever donated that would be identical to that lamb. So this is what we do when we do nuclear transplantation. Okay, but there's some problem with cloning like this. Well, one thing is the clone embryo has a lot of defects for some reason. We're still perfecting this technology, and it's really cool, and then we're learning a lot about it. More, it actually has something, because DNA, as recent scientists has discovered, DNA exhibits something very interesting. Well, every single organism exhibits this. It's called epigenetic changes. Basically, it's changes that happen with expression of the genes that cause a different behavior. So for example, well, not necessarily behavior, but cause a different trait, if you will. So for example, let's say if I have a clone, so Mr. Shu number 2, Okay, let's say we got separated, and then we are living in completely different life. Well, later when we come back together, let's say, oh, I found out I have a clone. Let's meet together and have a coffee or something. Well, when we meet each other at that time, we actually, even though our genetic information is identical, our expression of that genetic information will actually change throughout the year based on where I have lived and how I have lived and many decisions that I have. Basically, it's because on DNA, we learned this, DNA has different way to folding, so the chromosome doesn't have to fold certain way for genetic expression. And depending on that foldings, you can actually express or doesn't express certain part of the gene. So that's what epigenetic means. Okay, another thing really cool is stem cell. So stem cell have there's few type of stem cell. So you have zygote. So this one is totally potent. What that means is it can turn into any type of cell. Okay, and then you also have embryonic stem cell, which we'll call pluripotent, which means many different cell type. It can become many, but not all. Okay, and then lastly we have the adult stem cell. So multipotent, so a few cell type. So this is the most restricted cell type. But a lot of technology now we have the technology to kind of like force it become pluripotent. So this is the IPS process. So this is a breakdown of all the different stem cell. So basically you can sell the adult stem cell right here. It's this one, and then it can normally it can only become, let's say if it's blood cell, then it's blood cell. Okay, blood cell, stem cell. Okay, but we can actually look at right here. If we have embryonic stem cell, this embryonic stem cell has ability to transform all three. Okay, liver cell, nerve cell, or blood cell. Okay, so that's the big difference between them. But with some special technology, we can actually use stem cell, let's say this stem cell from the skin, okay, and then we actually undergo IPS treatment and make this one become pluripotent and then replace back to basically repair whatever tissue that's damaged. So by using the IPS technology, we can almost kind of help to repair whatever that's missing or whatever that's damaged. So it has a lot of promises. That's why we study stem cell researches. Well, these are some of the one more. These are some of the application of using DNA technology. We can also diagnose. We can do diagnostic treatment. We can do genetic therapy. We can do different kind of pharmaceutical production. We can also use it in forensics science, and we can also have environmental benefit. We can also have agricultural benefit. So let's get started. So this is a genetic therapy. So what we can do is, well, last chapter 19 we talked about using viruses entering different cells. Well, we can actually use viruses towards our advantage. By introducing viruses that has very specific genes, we can actually introduce the viruses into a specific cell, and then reintroduce that cell into human body. Okay, so by that we can actually either try to treat a certain disease or try to treat a certain symptoms like that. Okay. Also, we can, like earlier we kind of talked about a little bit, we can actually insert that genetic information and make certain animal to produce something that they shouldn't produce. So for example, this one, it's secreting specific milk that has human protein. Okay, that we can actually use when we extract that protein. Okay, so another thing we talked about is DNA fingerprint. I'm gonna save this for second part because, well, my recorder is running out. Okay, so look forward to part 2, and this is chapter 20, biotechnology, and this is part 1. Bye.